Hello everyone, welcome to the 10th and final INOTA webinar of 2024. INOTA is a cost action funded by the European Union, and it is the European Network on Optimizing Treatment with Therapeutic Antibodies in Chronic Inflammatory Diseases. I'm Edwin, I'm the leader of Working Group 2 on Individualized Treatment Optimization. And I'm really happy to host this webinar today. I hope you are all as excited as I am, because today we're going to hear everything about artificial intelligence and its role in precision dosing. For this webinar, we invited not 2, but 3 speakers. So let's get started with the first speaker, who is Dr. Nikola Stefanovic. Dr. Stefanovic is an associate professor at the University of Niš in Serbia, where he performs research focused on the optimization of drug treatment, with a particular emphasis on PK, so pharmacokinetics, and pharmacogenomics. His work is primarily centered on immune suppressive drugs used in kidney transplantation. With that, Nikola, I will leave the floor to you.
Oh, well, thank you, Erwin. Just to share my screen.
We can see it. Perfect.
Okay. Hello, everyone. My name is Nikola Stefanovic, as Erwin mentioned. I am associate professor at the Department of Pharmacy, Faculty of Medicine, University of Niš, Serbia. And in the next 15 to 20 minutes, I will try to make a link between pharmacometrics and model-informed precision dosing, MIPD, and artificial intelligence and machine learning. So since there is widespread of machine learning algorithms, I will mainly focus on supervised learning. Well, over the past 6 years, there is remarkable success in the field of precision medicine. Well, this success was pretty much driven by the advancement in computational and mathematical technologies. Therefore, we can define MIPD as the use of computer modeling and simulation to determine drug dosage, regimen tailored for an individual patients. This approach, well, should provide more favorable balance between benefits and risk compared to traditional approaches using the unique characteristics of the patients. Also, population pharmacokinetic, pharmacodynamic models, well, pharmacometric models are usually utilized in model-informed precision dosing to make clinical decisions for physicians. But these statistical and mathematical models, which underlies the pharmacometrics, became more complex, requiring significant computational efforts for large populations, especially when it comes to covariate selection, which is the most time-consuming part. Therefore, integrating pharmacometrics with advanced data science tools such as artificial intelligence and its subfield, machine learning, could be a transformative step in clinical pharmacology, enabling precision medicine advancement through utilization of big data sets. Also, I will mention one of the definition of machine learning, that is the field of study that gives computers the ability to learn without being explicitly programmed. Also, machine learning can be used to manage large sample size. Large sample size associated with big data provide powerful learning and predictive capabilities, hypothesis generation, and pharmacometric model development and improvement. Therefore, integrating those disciplines can provide a computationally efficient approach, which can process large and diverse data sets, which will enhance pharmacometrics in a sense that will supplement small sample sizes, which is typical for pharmacometrics, and this will expand parameter inference. Some authors think that we can assume that machine learning and artificial intelligence can be a bridge between, on one side, pharmacometrics, but on the other side, big data. There is various machine learning application in precision dosing, such as various application in precision dosing, such as concentration and exposure prediction, dose optimization, quantitative system pharmacology, MIPD system development, and etc. So Let's continue with machine learning. Well, we can divide machine learning into some sub-areas, such as supervised learning, semi-supervised learning, and let's say deep learning. So we'll start with supervised learning. Supervised learning, you use the data sets with the labeled outcomes to train a model to make predictions, regression models, or to make classification, classification models, but using the selected covariates as input for the model. Typically, regression models, typically supervised data sets in supervised learning can be divided into training data sets and test data sets. Important is that test data sets should not be used in any phase of a learning during the learning process. Also, considering some algorithms that are used in supervised learning, usually we use regression models such as linear regression, lasso regression, ridge regression in supervised regression models, all in classification logistic regression. Also, we use tree-based algorithms like decision tree, reinforcement, random forest. Then we used boosting machines, support in MIPD for, as I say before, prediction of exposure and concentration, personalized treatment plans, covariate selection, make distinction between patient classes on some predefined threshold, as we will see later. Besides supervised learning, I should mention unsupervised learning. It usually uses a data set without labeled outcomes. It is more focused on finding hidden patterns within a data set, which is important for exploratory analysis, hypothesis generation, and let's say which will inform downstream processes. Unsupervised learning is usually divided into clustering algorithms and dimensionality reduction algorithms. Also, I will mention reinforcement learning. You will hear more about it later, but it is a dynamical process of trial and error and can be used for real-time adaptive dosing strategies. So, what are the difference between pharmacometrics and machine learning? Let's say first the difference is in their objective. The objective of pharmacometrics is to characterize variability and assessment of parameters. Despite that, machine learning, the main focus of machine learning is optimization, prediction, well, to find the best model in which minimize the prediction error, so accuracy in prediction. Also, when it comes to pharmacometric modeling, there is always structural model, and which is based on physiological and pharmacological mechanisms, which allow the derivation of parameters which are clinically interpretable, such as volume of distribution, drug clearance. Machine learning is a completely data-driven approach, which removes the need for mechanistic assumptions. Also, the parameters within machine learning are more mathematical interpretable than biological interpretable. So machine learning has two main parameters, two kinds of parameters, let's say. First is hyperparameters, which are not learned from data. Hyperparameters are set at the beginning, can be optimized during some techniques, but they are usually they are responsible for learning process and the complexity and the structure of the model. Also, hyperparameters are usually closely associated with underfitting and overfitting. Let's say underfitting is the situation when the model is not able to catch the complexity of the data. So this model performs poor in training data set. Also, oppositely, overfitting comes when the model is, let's say, perfectly fits the training data, overlearns the training data, also overlearns the noise, and it provides poor performance into test data set, also with the new and unseen data. So optimizing the hyperparameters, we can minimize the overfitting. Also, machine learning models usually have internal or model parameters. Those parameters are learned from data, and they are, let's say, weights and biases in neural networks or coefficients in regression. These internal parameters are usually adjusted by minimizing the loss or error function, which is the base of predictive models, and also assigning weights to some features. We are directly responsible for the importance of that feature in the final model. So I have two examples. The first one is the article, which can be assumed as a proof of concept article of Koch and Associates, who demonstrated the possibility of applying decision tree classifier as initial step for covariate analysis. Therefore, we have two phases of this very cited articles. The first phase is creating a simulated data set based on the pharmacometric model, which describes the concentration by one compartment model with rate of absorption, rate of elimination, and apparent volume of distribution. So in order to create a simulated data set, we have covariate 1, covariate 3, which modulated the elimination, also have covariate 2, which influences the volume of distribution. So we have 500 simulated patients with 7 measurements and following time points. Pre-dose, 2 hours, 4 hours, 6 hours, 8 hours, 12 hours, and 24 hours. Also, we have those covariates, and in the end, we produce a data set which in a standard pharmacometric form with 10 columns, let's say 10 dimensions, like ID, time, amount, dependent variable, missing dependent variable, covariate 1, covariate 2, covariate 3. Also, we added two fake covariates, fake covariate 1 and fake covariate 2. In the end, because also we have one more not measurement, but a row because of dose administration, we have, let's say, 500 patients in 10 columns and 8 rows. In 10 columns and 8 rows, so we have 40,000 entries. So we create these data sets. Based on these data sets, we are going to machine learning phase. So we need to prepare a data set for machine learning. So we need, because this is a regression, we need a labeled outcome. So the labeled outcome is about what we want to do, what we want to prove. So we have 2 research questions because we have 2 parameters, one with elimination, one with distribution. So we have 2 questions. First one is, what are the risk factors that the patient will have a half-life higher than a given threshold? So we define the threshold, predefined threshold is, for example, 7 hours. So patients who have more half-life above 7 hours have labeled 1. Those one with below 7 have labeled 0. Also, with the second question, what are the risk factors that a patient will have a drug concentration, maximal drug concentration above given threshold? So we have authors provided predefined threshold is 3.5. So patients with maximal concentration above have labeled 1. Those with below have labeled 2. So after that, decision tree was trained within entire label data set. The authors did not split a data set on the training and test set, so because they do not have prediction. And the results show is shown in a context of feature importance, where covariate 1 is 65% in 65% the most important, and covariate 3 27%. This is pretty much in accordance with pharmacometric dataset. Also, the same situation with another dataset where covariate 2 is in 78% the most important feature. What I want to note here is that this dataset using for machine learning is pretty much smaller, because we only have, as you can see on the table, 6 columns, because we don't need, in this case, ID, we don't need amount, we don't need time, we don't need, let's say, missing dependent variables, so measurements. We don't need missing dependent variables. We only need covariate 1, covariate 2, covariate tree, fake 1, fake 2, and the column with the labeled outcomes. So this data set has 3,000 entries, which is quite, which is less than a classic pharmacometric data set. So we have the same result in the end, but this approach is more efficient, and faster than pharmacometric. The another example is about integrating artificial intelligence, in this case machine learning, in TDM through construction of a concentration and exposure predictive model. So I will use the work of Boilard and Associates, who developed extreme gradient boosting models to estimate tacrolimus interdose area under the curve, AUC, based on the limited number of concentration. In this case, 2 or 3 concentration and some other predictors. So the model performance was compared to maximum a posteriori Bayesian estimation, and these models also compared in external validation data sets. This is a data set, 6 full PK data sets from which AUC was calculated based on the traditional trapezoidal method. So, development, 2 for each of tacrolimus formulations. Tacrolimus has twice a daily formulation, which corresponds to AUC 0 to 12 hours, and have once a daily formulation, which corresponds to AUC from 0 to 24. Also, we have models with 2 and 3 concentrations. When we use HGB, extreme gradient boosting model is, in this case, ensemble regression model, just to mention, which is based on the small regression, on small trees with the regression models within. So when we use 2 concentration models, we use trough concentration and concentration 3 hours after dose administration. When we use 3 concentration, we use trough, C1, 1 hour after dose administration, in 3 hours after dose administration. Also, as feature, we use morning dose, tape of transplantation, age, time between sampling and the transplantation. Also, so now we have the complete data set. Data set is divided into training data set, 75% of all of the whole data set, and the test data set. The training data set is used to build the model, tune the hyperparameters, and evaluate model performance by, in this case, one technique, tenfold cross-validation. So what we have 2 rounds of tenfold cross-validation. First round of tenfold cross-validation is used to tuning hyperparameters, such as learning rate, such as the depth of the tree, the number of data points in a node which need to split furtherly. So when we finish, when the model, when the hyperparameters optimized, then a new round of tenfold cross-validation was performed, and we evaluate the relative importance of predictors, in this case features. Also, the full model were optimized by searching the best combination of internal parameters, which were evaluated using these cross-validations and assessed by using the mean, root mean square error and R squared and their standard deviation and scatter plots. So this is the usual metrics that are used in regression prediction models. When the best models were chosen, they were evaluated in the independent test set, and the same metrics were used. So as we can see in this table, these models perform very well because the relative random mean square error is below 10%. What is important here, that we have pretty much same results in a training data set and test data set, which can suggest there is no overfitting. Also, when those models were compared in external data set, they perform very well, better than MIPD models. Also, in some cases, models based on two concentration were better than other models. So we have mostly random mean square error below 10%, also bias below 5%, and also very much small number of profiles out of acceptable plus minus 20%. So in the systemic review published this year, Lee and Associates given a site about publishing articles on decision dosing using machine learning methods, and they indicate let's say the significant increase in the number of published publications during 2021-2022. This has been continued later on, which can be seen on the PubMed search from the PubMed search results. And I want to say that most of the models, most of the articles consider precision dosing in immunosuppressives, anti-infectives, and anticoagulants, well, mostly in drug with narrow therapeutic index and variable pharmacokinetics. Also, most of the researchers used retrospective data, but real patient data, and most of the methods which are used are supervised regression models and neural networks. So in the end, I want to make some future directions. Let's say we should focus on sample size requirements, not only on the quantity, but also on the quality of label data set. Also, machine learning can be very important in exploratory analysis of high dimensional data where we have small sample sizes, like in omics results from omics technologies. Also, efforts should prioritize identifying findings consistent across multiple ML algorithms. ML is not replacement, rather complement to population pharmacokinetics and pharmacokinetics pharmacodynamics. Also, more profound use of electronic health records because of there is vast amount of structured and unstructured data, but also integration, real-time changes, dynamic prediction. External validation is needed, and also clinical relevance in a context of efficacy safety. More clinical interpretable features should be used in models. Also, we should focus on ethical concerns. And the conclusion, I will brief. ML algorithms may have a transformative impact on pharmacometrics, but their clinical utilization is in early stages. Integrating pharmacometrics in machine learning could enhance prediction accuracy, support treatment individualization, improve clinical outcomes. Collaborations with large network groups are needed. Also, generalization, external validation, and clinical utility assessment using real-world data are essential, particularly for models which are derived from simulated data sets. Thank you.
Thank you very much, Nikola, for